So next we have a group discussion from a group of panelists. So here we have Mr. Elia Brodsky, the co-founder and CEO of Pine Biotech. We also have Dr. Raja Guthrakuta, a professor of astronomy and astrophysics at UC Santa Cruz. Dr. Japoliano Chavez, a postdoctoral scholar at the University of Chicago. And Miss Maria Victoria Luz, a bioinformatician from the University of Brasilia. So they are going to be discussing the SIP program, how they all got involved in the field of bioinformatics, and if they have any advice for new bioinformaticians. So please welcome.
And thank you, Laura. Before we jump in, I know that Raja, you're going to introduce us to the SIP program, but before we jump into that, I just wanted to highlight a few takeaways so far from all of these wonderful presentations. So in the scope of today, we heard from people in India, in Africa, in South America, in the US, people that come from life sciences as well as computer sciences. And we heard how there is a common challenge of introducing, because this topic is kind of central to our symposium, introducing bioinformatics at an early stage. Many people don't realize the significance early on, but later on, as they continue to pursue their research interests, they kind of rediscover the significance of this skill set and go on to apply it to a variety of topics. We saw people that apply it to studying insects, studying plants, bacteria, eventually pursuing cancer, biotechnology, big data, cloud computing. So it seems like the trajectories are very unexpected. But what they all share in common is that early exposure to bioinformatics, appreciation of the role of data in biology, and eventually learning how to utilize these skills to pursue what they really enjoy, what they love, what they care about. So that really emerged to me as a common trend. Another important trend that I saw was really the diversity of what we call bioinformatics. I mean, it ranges from cloud computing, like I mentioned, all the way to mathematical modeling, like we just heard right now, to analyzing kind of simple data and very, very complex data, right? So it's really a term that encompasses a diverse set of directions and challenges that it seeks to address. And my final takeaway from this is that there are so many efforts to try to introduce these topics early on. And a major question that arises for me is, yes, we are here because we're all interested in bioinformatics. What about people that maybe are outside of the circle? You know, people that don't necessarily, tend to enjoy research or pursue research. You know, how do they get exposed to these things, and how do we make that early interaction fun, meaningful, and leading to something effective later on? So with that summary, Raja, over to you. Let me just make sure that you can unmute yourselves. There you go.
Oh, thank you. Thank you very much. I know Laura mentioned that I have about 12 minutes to speak and a couple of minutes of Q&A after that. I have a slide set. Thank you for inviting me here. I'm an astronomy professor at UC Santa Cruz. What am I doing in a biotechnology research symposium? Let me explain. I got connected to Gio Chavez, who was a grad student at UC Santa Cruz when we got connected, because we were involving high school students in our research. Gio connected me to Elia, Pine Biotech, and to this Omics Research Symposium 2022. I have a few slides to share with you. So let me see if I can share my screen. There we go. Are you able to see my slides now?
Yes, we are.
Okay, thank you. I will talk not so much about SIP, but the CREST umbrella of programs and initiatives, and I'll tell you what CREST is in just a moment. I'm a professor at UC Santa Cruz. I have the misfortune of being department chair. I can't wait for that to end. And I've been running the science internship program for high school students. And 7 years ago, I spent 8 months at Google, and they really helped me scale up some of the outreach work I was doing. This work has been done in collaboration with my colleagues at UCSC, Sarah Schneider, Christy Tall, and Carolyn Roosevelt. What I'll talk about in a moment is CREST, which stands for Creating Equity in STEM. Santa Cruz is a beach town. I hope you can see the shape of the crest of a wave here, but it's also the letter CREST. And it's an umbrella that is, that sort of brings coherence to a range of things we're doing here at Santa Cruz. And I'm really grateful to you for allowing me to share this. This is, our bold title is Creating Equity in STEAM, where STEAM is science, technology, engineering, art, and mathematics. Are we going to create equity in my lifetime or yours? Probably not. Are we going to create equity with one person working on this? Probably not. But the whole idea is for us to join hands, and that's what this symposium is about. Join hands, work together, and work towards this thing. This is a bright light in the distance, but it can guide us in our work, and that's what I'm hoping to talk about. Why do this? There are folks from India on this call, so I thought I would mention something about the Indian cricket team. I'm a huge cricket fan, and the Indian cricket team in the 60s and 70s, when I was following them, were constantly losing. In Brazil, on the other hand, the Brazilian soccer team was constantly winning. There was some difference in the formulae. The Indian cricket team, when after every loss, the captain or the best player on the team, the person who had put in their best performance, would be interviewed by the television or newspaper in those days. And even though they had all grown up in India, they spoke the Queen's English. They spoke perfect English, growing up in India, playing cricket. Now, there's something wrong with that picture, right? Because you don't need to speak English with a perfect accent in order, the ability to do that and the ability to fling your body into the air and pluck the ball out of thin air with your hands, or to good hand-eye coordination, that has nothing to do with the ability to speak English. So this changed for the Indian cricket team in 1983, where for the first time they had a captain who could not speak a single sentence of English. He was an embarrassment in front of the camera and the television. They had translators for him to translate from his native language, his native Indian language, which was Punjabi, to English. But guess what? The Indian cricket team won the World Cup in 1983 with this captain. I don't think this is a coincidence, right? When you open your talent pool so that you're not just selecting people who can speak perfect English and they're part of an elite section of Indian society, when you open your talent pool wider than that, you're going to get better talent. And the Brazilian soccer team realized this a long time ago. If you look at almost every Brazilian soccer star has not been from an elite background. Almost all of them. Very few have been from elite backgrounds. So diversity and excellence are not two separate things. They go hand in hand. Other thing I realize is we are working to diversify our institutions, but that requires working outside the institutions. We can't just expect that people who will apply to our institutions as undergraduates, graduate students, or faculty are going to be representative of society around us if we don't do the groundwork, if we don't bring ourselves, our university work, to the outside world. And this is true for corporate organizations as well. We have to work outside to diversify the inside. And I want to say that we are going through a pandemic, COVID-19, but we're also in an information pandemic where the value of evidence-based thinking, the value of the notion of expertise has really eroded. And while there's a vaccination, I just took my 4th shot yesterday, my Pfizer shot for COVID-19, and they give it to the elderly first, because that's who's at risk. For the information pandemic, where we've eroded critical thinking, the people who are most at risk are the young people, because they're going to live with this disease the longest. So we need to address that, and programs like these, like Gio and I are involved in, are the vaccination for young people. And it's a long game. It's not a short game. It's not something that's going to address the immediate needs of the community. It's not going to immediately address hunger or domestic abuse, poverty. It's not going to address those things right away, but education is a long game. Okay, with that, I'll get off my soapbox and talk a little bit about like a pattern I see. When young people are going to engage in STEAM, the most logical thing for them to do is first gain access to the people and the process. And I've underlined people and process here, really, because very often when science is reported, you see the gee whiz results. You don't see the many things that fail to get to that. So people and process are important. For me, that access happened by going to a planetarium as a kid or by watching David Attenborough's Life on Earth. Then you need to build the skills, and then you can apply them in a controlled setting where the mentor has already gone down a path and they send other students down the same path, where they can control the amount of instruction they give them. And then finally, there's open-ended research. And the thing that Gio and I worked together on in SIP involved open-ended research. By open-ended, I mean the mentor has not solved the problem before. There's no recipe to solve the problem. And you're engaging young people in that process of learning from productive failure. So creating equity in STEAM is something we're trying to do. And the words that run across are what we're trying to achieve. And the words that run up and down, critical thinking in the context of open-ended research under close mentoring, is how we're trying to achieve it. So the goal and the method, goal across, method down. So we first want to encourage young people to dip their toes in the water. This is the access piece. And I've been running something called Shadow the Scientist. Now when we get time on, I'm an astronomer, so I use telescopes in Mauna Kea, telescopes in California. Instead of going to the telescopes, we can't go to the telescopes anymore because of the pandemic. What we do is operate the telescopes from home using VNC, using Zoom to connect people. And what we've started doing is encouraging young people to join our Zoom call to see how we work. This is dipping your toe in the water. This is seeing, do I like the temperature of the water? Do I like the people who are already in the water? And then if you like this, then you have to learn the swim strokes. And this is something we've been running a Python and research tutorial where you learn the techniques. Gio has started this wonderful R in the context of research in computational biology. The idea is to teach young people the tools, the computational tools, and really embedding this in research. What we do here is we have code that's directly relevant for a science project, and you can remove lines of code and ask the student to write them back in to make the code work again. And what you're doing there is you're teaching them both about coding, but also about the application of that coding. Once you have this, then you're ready to take a deep dive. That's what the science internship program is. It's open-ended research, 10 weeks. Today is our application deadline for our high school students. So it's too late if high school students are interested. Today is the deadline, but we have over 2,000 applicants. We have a second program that's in collaboration with Professor Annette Lee at Arizona State University. That's at the intersection of art, science, and culture. It's a different kind of deep dive, not just into one science, but into an intersectional area. Then once you do this program, after this plunge, what do? We have a network called the SPHERE Network, which stands for STEM Programs for High Schoolers Engaging in Research Early. It's a way to connect programs so that we're not each reinventing the wheel. It's a way to create a community of practice for mentors who engage high school students in research. We have an alumni buddies program, so near peer mentoring. We follow the trajectories of our interns. We offer this to teachers in the science internship program. That's what TSIP is, so that they can take this work and spread it to their students in schools. We have a peer-to-peer college and career network, and we have community college students in SIP. So that's the end of my slides. That's my 12 minutes. I'll stop here and see if there are any questions. Thank you.
Okay, I actually do have a couple of questions. So the first one is, what is that critical thing that can lead a student to go from following a tutorial to starting with open-ended research?
I can tell you what I've found, but others should chime in. I found the first time I was doing research was when I was a grad student. But for younger people, for high school students and college students, what I've learned is the first time they're participating, when they feel like they're, if not equal partners, but at least partners in the enterprise. When the mentor turns to them and say, what do you think of this part? Why are we seeing this trend? That's a critical moment. That's a critical moment where you know that you're not just doing this for someone else. You're being asked to think about something. You're close to an equal participant in this. That's what our students have told us. That when we felt like we were participating, but in a way that where our opinion was valued as a scientist or engineer. That tipped the scales for us. That gave us the confidence to really feel like we have ownership.
I think in other words, Raja, a good way to put it is the feeling of community, feeling of being part of something. Maybe my way of putting it is to feeling that you're part of something bigger. For instance, the one interest that I have that motivated me for doing this type of research that I do is the possibility of curing people, of helping people get cured for the diseases that they face. And so that's my motivation. That's the one thing that makes me never give up on the drawbacks that I kind of face daily on a day-to-day situation, because the result of the effort that is going to be put is going to be much better than the whatever is costing. So I think that from the beginning, not from right now, I think the opportunity that I had on SIP, and that causes me to strongly appreciate that, is the fact that this experience of belonging that I have as a mentor can be shared with the students that come to join the program. And because of the philosophy of SIP, which is in my mind is a thing that you have taught us, you have pioneered, because of the philosophy of SIP, that becomes something that is offered to the students. I mean, this mindset, the possibility of feeling the belonging to the reasons, to the cause of research, is passed on to the students very clearly, in my opinion.
Maria, did you have any? Sorry,
yeah, please go ahead, Maria. Maria, please go ahead.
Yeah, and I'll just briefly mention that Maria is a student. I think, can you unmute yourself, Maria? Yeah. Oh.
Hi, everyone. I am Maria, and I'm a student of biotechnology in Brazil at University of Brasilia, and nice to meet you all. And I will put my presentation here right now.
One second, Maria, we will get to your presentation. I was wondering if you just had anything to add about, how does a student go from just learning, doing what I'm told, to participating in some research? What do you think?
Oh, my experience was very different because I had classes at university about bioinformatics, but it wasn't my focus at all. I wanted to study plant sciences and everything, but then I started to compete with synthetic biology, and I faced this challenge to learn by myself all the tools that I haven't learned at university. And it was a very interesting moment, of doing things by myself and going to the internet and researching. And then I tried to talk to professors that are experts in the area, but it was a very challenging experience in bioinformatics. And it's still hard for me.
Yeah, I wanted to add, since we are coming from the same background, because I didn't have any bioinformatics at all as an undergraduate. So I didn't even have the inspiration, the where to go to about the bioinformatics. But as you said, if you either if you're exposed to or not, I think that motivation that drives is what really can get you to accomplish the goal. So you said that from the beginning you were interested in plant science, which for me, I was really not interested in plant science. But now we come to the same place. We're interested in the same thing,
field.
Exactly in the same field. And this brings something that I learned a little bit with Raja, which is we are from completely different backgrounds. He's from astronomy. I'm a biochemist. These are very diverse, complete in the sense that he is looking to the cosmos and I am looking to the microscope. these are very opposite fields, but at the same time, we come to this area where the knowledge of programming and of coding completely intersect.
Okay, so Gio, did you have slides that you wanted to share?
Yes, if I have the time to.
Okay, so let's do this briefly. I have a few more questions. Yeah, go ahead, Raja.
Sorry, I was just going to say one very quick thing to address your point, Elia. I think the way we train our students, they're trained to self-betterment is what they're, if you do well, if you study, you'll do better at this exam. Self-betterment is how we teach in schools and colleges, whereas research is about betterment of the world. It's not about improving yourself. And I think that's a very important switch.
I have to say, you have very interesting insights. Both I liked your cricket analogy, and this was also very interesting. Thank you.
Thanks.
So I should
okay,
just go ahead and...
Yes, it's all yours. Yes.
All right, so I guess I gave the title of my talk, Computational Programs to Understand and Heal Diseases, and I guess I already gave you an introduction for that. Why do I hold this worldview? So one thing that happened in Brazil very recently, and this motivates a lot of my personal engagement in this whole story, I think, is the fact that the federal government just announced the intention to sequence, I think their number is 1,000 Brazilians. And the reason for that is because the government now wants to understand the genetic backgrounds of the Brazilians and how that can inform the public health system. So for a Brazilian, I grew up with this notion that Brazil is an unequal country, that there's a lot of need in terms of resources. So this particular event here for me is really emblematic, because we are now at a situation where we are thinking about sequencing the people, but honestly, we need to develop ways and tools and human resources to allow us to access that information, understand that information, and make a good use of that information. So I think here we are talking about fields like big data. My field is the field of DNA sequencing. And we are basically talking about how can we make this information benefit an entire society, or as much people in a society as possible. So we have this notion that there is a lack in representativity in many sectors of societies, in many groups of societies. But unfortunately, an inequality is present everywhere in life, and it's a fact of life. Even in the USA, which is the richest of the countries, there is inequality. And I also learned this when working at the science internship program, because what we do in the science internship program is we try to help as much students with problems in their resources as possible. And also in the United States, several institutions deal with the same problem, and they, in many cases, they do the same type of approaches to the problem. But I don't want to talk about a lack of representativity that may be a little bit subjective. I want to bring the attention to this field, the field of genetics or genomics, because I was reading this very interesting paper that stated that around 10% of the human genes, or 10% of, you can argue as 10% of the human genome to be well studied, and for instance, has allowed us to develop drugs to treat, let's say, only 10% of the conditions that come out of genetic problems. And so this statement that I am highlighting here in yellow is a state that this group published, And basically they concluded that in 2015, 50% of all the publications came from only 16% of the known genes at 1991. And the information that needs to be addressed here is the fact that we were only able to study these genes because there was research going on. And even though there was research going on, there is this inequality in the amount of research that is done for some groups of genes, and other genes are not researched at all. That happens possibly, I want to mention, because we just didn't have the time or the investment in researching all of these other genes. But again, we come to the same point, which is the lack of representativity of all of the genes or all of the genomic regions that are present in the human genome. And because of that, just because of that, some people are going to lack on the possibility to have their health problems cured or treated. However, there are legitimate reasons for this lack of representativity in many of these studies and in many of these areas. So here, first I want to acknowledge this problem that we have in the genomics field, and then I want to address that from the perspective of the two diseases that I have studied during my PhD and now during my postdoctoral study. One thing that I noticed we for sure need is innovation. We need to think differently. We need to think out of the box about all of these problems. And in my experience, building the community is something that is not necessarily innovative, but I think I learned at SIP that that's the innovation that we kind of need. So now, moving a little bit to the diseases that I studied. So Huntington's disease is actually what we call a monogenic disease. It's a disease that is caused by a condition in a single gene. This gene, I haven't put it here, but the gene is called Huntingtin. And the gene, it's a neurodegeneration that is caused by a CAG expansion. This CAG, for those of you who may not be familiar, is a sequence of bases that are expanded in this condition. And that expansion in the DNA causes the protein that is encoded in this gene to have some different chemical properties. And that's the source of the neurodegeneration in Huntington's disease. Because the genetic of Huntington's disease is so simple, I would say simple in the sense that it is one gene only, and that this one single gene has this dramatic impact in the health of a person. It was well studied in the 20th century. And then what we had was the Huntington's disease was actually one of the first diseases for which we had these genetic consortia that were established in the past century. On the other hand, a disease like neuroblastoma is a polygenic disease, which means many genes, many factors contribute to the establishment of a pathological condition. There is one example of this MYC-N amplification that occurs in neuroblastoma, but I'm not going to talk a lot about that. So here I'm showing kind of where the disease happens in the body. So with this comparison of Huntington's disease and neuroblastoma, it is what I want to say is that it is legitimate for this lack, let's say, of genetic consortia to study certain types of cancer, not compared to Huntington's disease, because remember that I said that the genetic consortia for Huntington's disease were established much before cancers, because studying the genetics of cancer is more complex in the sense that there are multiple genes affecting the condition. In both cases, both in Huntington's disease and neuroblastoma and cancers, this genetic information needs to be stored. And when I say needs to be stored, I am talking about the sequenced genetic information. The sequenced genetic information, the genetic information that you know is written in the human DNA, it needs to be stored. And I think that there is a gap in the availability of programs that allow people to have that experience where they're going to learn how to analyze this genetic information. And I think that's how SIP and the programs that in Pine Biotech also, Omics Logic, fill this gap is by providing the tools, the environment, the community that is required for people to have that exposure, to have that experience. As I mentioned, I didn't have any bioinformatics background as an undergraduate student. And in my mind, that has not changed. There is some minimal exposure of undergraduate students to the field of bioinformatics. In many cases, that's not a mandatory exposure, but I feel that I think I cannot emphasize enough how much I believe that that exposure is completely necessary, because DNA is information which can be analyzed through computational algorithms and so on and so forth. So that's the motivation for the SIP and the Recombio outreaches that I have been involved. This picture here is showing an outreach talk that I did at the Instituto Federal Baiano, from which I am an alumni. So we have these. It's also a set of tutorial, but really here what I do and what I try to do is to help the students have a sense of what installing a program is, what running that program is, and what does it really the student in the Recombio approach, really the student has a feeling of what it is to interact with the machine and install programs and then starting doing their analysis from scratch. I think that's something that I value a lot, doing the analysis from scratch at this point. So as far as the mentoring perspectives in SIP and Pine Biotech, that's completely valuable, but I really value also the possibility of people engaging and understanding how the research that we do is important. And here are some illustrations of that at the SIP program. The other thing that I think is very useful, and I guess I think I now want to address a point that Elia brought as far as why do we want to go that route. So I didn't mention to you, but my mother was misdiagnosed with an infectious disease when she actually had a cancer. So clearly at that point, there was a problem either with my family or with the medical system or with the government that financed the medical system, because just by not having the appropriate diagnosis, there is some problem going on with either the educational system or the medical system. That's one of the reasons why, in my mind, we cannot really differentiate education from medicine, because we need to have education is the process that allows the development of the tools that are going to enable us to treat people. So what I'm trying to say is that in a situation like this, where you have a diagnosis problem running in your family, you need to I was faced with the problem that not necessarily we have the best diagnostic systems in the world, and if we have it as good as it possibly could be, we also needed to make that diagnosis available to as many people as possible. So this is why I think I need to mention this idea, this concept of liquid biopsies, because in neuroblastoma, what happens is that the tumor needs to be accessed in the patient, but in the other hand, you need to have certain growth of the tumor in order for you to have access to what's going on with the tumor and the cells. That's why with a biopsy from the blood of that patient, we get a chance to look at many other interesting biomarkers, I would say. So here I am bringing the examples of cell-free DNA, cell-free RNA, nucleosomes, exosomes, cancer-free cells. So we can basically collect these markers, these biomarkers from the blood of a patient, and we can basically interrogate, we can basically study, we can basically understand and educate ourselves about the progression or the biology of these diseases. And my understanding is that you are not going to understand that if we do not understand how to do the analysis, if we do not understand how to interpret the genomic data, how to collect the genomic data, how to analyze it properly, how to develop the strategies that are necessary to treat the patients, to diagnose the patients properly. So this is why I think that understanding the bioinformatics tools is at this point in time, I think it's crucial. I think that in the future, at least, obviously, the societies that are more financially privileged, we're going to have these biobanks that store information on the blood of people, on tumors of people, and this is going to be very useful information to have and to have access to. And there's going to be many opportunities in my mind to study that and to help treat these conditions in people.
So, Gio, I'm going to just jump in here. We have a few questions for you, sure. But for the sake of time, I think that, this is a very critical topic for us to continue and explore, and I'm sure that we will have the opportunity to do this soon. But as a way to kind of bring this together, Maria, similarly to yourself, right, is a student that got exposed to bioinformatics almost by chance and has a very fascinating story of how that eventually turned into something very practical and meaningful to her. So before everyone leaves, I wanted to give her the chance to share her story briefly. And then if we have a few minutes at the end, we can address some of the questions that we already have from the audience and kind of bring your story to connect to what she has to say. Is that okay?
Sure, absolutely.
Okay, thank you. Okay, go ahead, Maria.
All right. So I am, as I said before, I am Maria. I am a biotechnology student and I work as content writing intern at PI in Biotech. And I will tell you part of my relation to bioinformatics here and a little bit about my history and also give an example of how we should explore this area being undergraduate student or college and high school, for example. So bioinformatics, it was part of my underdegree curriculum, but Professor Papas was like one of my biggest inspirations because he teach us how to learn about NCBI, protein data bank, this kind of things that are right now very basic, but this is my way, the way that I start to learning bioinformatics. And but my biggest goal in university was study plant sciences, as I was saying before, specifically phytochemistry and work with the biodiversity in Brazil. But I wasn't aware that my degree was so much than this. I work in communication, in administration, and in bioinformatics. So then appears this great opportunity to create and to lead an IGEN team. So our goal was to develop a microbial fuel cell to generate electricity from leachate. And this was beautiful passion of mine because leachate is a local problem of Brazilian where I live, and the whole problem was this leachate leaks, and we wanted to treat these persistent pollutants there and also take into consideration the other that are present in the city because of the Brazilian sanitary landfill. So this way, we listen in also the Brazilian sanitary landfill and the population. And bioinformatics was used a lot because the whole project was in silico. And I wasn't so familiar with the tools that we need in the same way as my teammates because they had different backgrounds far away from bioinformatics. For example, my friends are from pharmacy, agronomy, administration. So to achieve our goal, we construct a genetic circuit in silico. We start learning independently, and that's the message that I wanted to send to you, because you don't need a degree in bioinformatics or a post-graduation and master's degree to enjoy the benefits of bioinformatics. But you have to be proactive to go ahead and learn something by yourself and discover your passion. And then after you discover your passion in science, you can introduce bioinformatics. So we applied computation in synthetic biology, in environment, in energy, because our goal is to generate energy using microorganisms to poor communities in Brazil, because we were born and raised in low-income families. And we also took into consideration like the social part. So we produce materials for awareness about the environment. And this was also a point that made us to be winner of gold medal in this competition. IGEN competition is like the biggest competition of synthetic biology in the world, but we participated in this version from Latin Americas. And this was a project that was very important to us because we wanted to bring for our people things that are worldwide, you know? So we became the first Brazilian team and the only Brazilian team that was finalist in this competition. And we also won this best project in energy, environment, and biodiversity track. And we were nominated for best human practice and local hero. So we saw how important was bioinformatics for our community. People that don't even know what synthetic biology is, what a molecule is. So we bring all of these things together to make our place a better place to live. And because of that, we achieved many things. We had many opportunities to present our work, including at the campus party, which is the biggest geek fair in the world. And the last few days, there was a version in Brasilia. So we also won some incentive grants and scholarships, and we are now changed like forever because we continue to learn each day a little bit and about this field, such as the omics logic courses that I began to watch and to study. So I recommend for all of you to start in some place, even if you don't know where you're going to apply this knowledge. But when you find the right place and the right opportunity, you will use bioinformatics. I am 100% sure. So this is my contact. You can talk to me anything and ask me some question, and that's my presentation.
Excellent. Well, thank you very much. We're already way over time. We have scheduled this to end at 4. So, taking additional time from all of the people that participated. But I do have an important question, and I want to start with Raja. You bring years of experience to this. Why is it that the academic world is so appearing to be maybe closed off to people that are just coming in? Why does it seem to be so such a big gap between people that are doing actual serious important research and students that are just coming up and getting involved?
It's a great question. I don't know. I think I don't think there's a proper recognition of the amount of brain power. I think it's two things. I think it's tempting for us to think that you have to have gone through all of these years of training. It's tempting to think it's a linear process. It's not. People talk about the STEM pipeline. It's not a pipeline. You can plug in at any point, right? You can, the work I do most of my day can be done by a trained monkey. No, that's the sad truth, right? It's very possible for young people to do this work without having gone through this long winded training. That's a sad truth. People don't like to acknowledge that. I don't know why. They don't like to acknowledge it. And I think they're not aware of the sheer amount of brain power in the, let's say in the, 12 to 20-year-old or 12 to 18-year-old, population in the world. It's taking a person from that self-betterment mode to world betterment mode is hugely motivating. You just have to reorient that person's thinking and they become a powerful force for good when you do that. I've seen this happen myself, but I think more and more people are turning to it, Elia. I don't think it's a lost cause.
Excellent, thank you. Well, my second question is for Gio. Gio, you mentioned that Brazil just has this goal of sequencing 1,000 genomes. Just today, or maybe yesterday or the other day, there was an announcement that the All of Us project has released 98,600 genomes that they sequenced in the past, I think, couple of years. So, the goal of the All of Us sequencing project is to introduce more diversity. And I think 42% of the people were white, as expected. So what do you think the world is missing out on when the numbers are so disproportionate, between countries that, have such efforts and countries that maybe do not have as large numbers as you showed in Brazil?
I think that, for the Brazilian part, I think that the actual interaction between the diverse chromosomes that compose humanity, that there is a peculiar case for Brazil because there is high degrees of diversity in Brazil because of the colonization. We had this admixture of these different populations, mainly notably from Europe, Africa, and American Native Americans. Of course, there is a matter of income. Only those countries, I would say, that have the resources to afford this can do it. And for good or for bad, that's going to be these countries with huge populations like Brazil, the USA, China, and India also. But certainly the other populations that are underrepresented in the world, with smaller populations, like small countries in Africa, small countries in Asia. There's representation of those populations in other countries. In many cases, that I know that to some degree, these genetic consortia, they do what they can to increase the representation of these populations. And unfortunately, in many cases, you're not going to be able to see the interaction of these chromosomes as you are going to see in Brazil, but in a way or another, this information can be available, or this can be analyzed. I think one thing that is necessary is the interest to analyze, and by interest, not necessarily I am meaning the interest in only investing in this type of studies. I am meaning the interest for people to go out there and download the files and put their hands on downloading these files, which per se is a strong effort to do.
Excellent, thank you. My last question is for Maria. So Maria, I know that you showed us you do like a million different things, right? You write for different companies, journals, you know, you have like 10 different jobs. What is the one thing, you know, if you didn't have to, you know, what would be the one thing that you would apply yourself to if, you know, you could choose from anything? And what do you think would be the benefit of just that single focus if you had the freedom to do that?
Well, my purpose in life actually is work with bioeconomy. So I want to help other companies with my knowledge to make all of different parts of the world more sustainable. And I think that all of the tools that I am working on and all these jobs, like in pre-acceleration of startups or bioinformatics or communication, are going to be some like of soft skills for me to make the world like a better place to live, you know.
All right, great. Well, it was a pleasure talking to all of you. Thank you again for joining and sharing. I think there's so much that we can get from all of your conversations. And I think if we had just a little bit more time, we would learn a lot more. I want to briefly mention that tomorrow we have our second day that's actually dedicated to industry and practical solutions, right? And so what I mean by industry is we're going to be hearing from people that work in biotechnology industry, in pharmaceutical industry, and they're going to share their insights about the role of bioinformatics and the significance of bioinformatics in that field. We're also going to hear about novel ways to use and simplify bioinformatics so that it's much more accessible and much more easy to interpret the data. And so if you're planning to join, we're going to have people like Anka Maria Ciobano from the Pistoia Alliance from Massachusetts. She's going to be speaking about Pistoia Alliance's new Women in STEM Leadership pilot program, as well as their program on the efficiency and effectiveness of R&D for big pharma. We're going to have representatives from the Tower Bioinformatics Research Center speaking about innovation with cloud computing and different types of analyses. We're going to have several speakers from NASA, from the Blue Marble Space Institute of Science to talk about astrobiology and what's happening there. And we will have prizes for those who presented today in the poster session. So we're going to have prizes, so be sure to join that if you presented. We're going to have some more people speaking from the National Association of IDeA Principal Investigators, Google, from the Precision Medicine Lab here at Louisiana State University, and also speakers from the Puerto Rico INBRE program and from the INBRE in West Virginia. So again, thank you everyone for joining today. Thank you, Raja, Gio, and Maria for your insights into this problem, and we'll see you hopefully tomorrow.
Thank you, Elia.
Thank you so
I
much.
learned a lot from you, Maria and Gio. Thank you.
I too learned from you. Thank you.
Take care.